So the patterns of inheritance is an important topic to understand, and it goes well with the sequence of topics that we have seen, such as meiosis and how we get the genes from both parents, and through this gamete formation and fertilization, producing the egg cell and the sperm cell. Each of them have a different combinations and create variations. Okay, so the prior to this one, there were a lot of misconceptions of about inheritance, and the misconception is that only males contribute to genetic material. So this is, again, I wouldn't say this is a worldwide confusion, but this is some pockets of people were confused about this. So they thought there is a little human being in the sperm cell, and the little human being develops into an adult, and even they had a name for this called homunculus. And so the egg provides a nourishment for this little human being, and then baby comes out. Okay, so that was a kind of a dumb idea, but people thought like that. And the other idea was that there is blending of genetic material, from both parents. So in this case, how can, why this could be correct, but you need to understand blending means like mixing together to create a combination of both. Like you mix a red paint and a white paint, you get a pink paint, like that. So you cannot separate the red from the pink after you mix it. So that's another misconception. So the work was done by Gregor Mendel. So he was working in a monastery, and he was not a professional scientist by any means, or a professor working in some university. So he was in his monastery doing experiments with pea plants. So this is a very short time experimented with pea plants to determine the patterns of inheritance. Okay, so this is something easier to do relatively compared to breeding animals and dogs that people were doing by crossing animals and crop plants, but they didn't know what to expect. Okay, so this is something that he was working on, and this is all in like 1860s. Okay, so that's a time point that he was working around this, and he published the result at that time frame, 1860 to 1880 or 90, but people did not understand. He used a quantitative approach in terms of understanding, how many offsprings had this characteristics or that characteristic, and also selected a good model system, which is the pea plant, which was easy to grow, easy to cross and hybridize, and also he selected the characteristics characteristics, so which are like a flower color, okay, or seed shape. So this type of characteristics that are determined by single genes. So these are some of his, secret recipe for Mendel's success. So he did the experiments for only about 8 years, and then he published all of them, and he was done. He was gone, and late 1800 or 1900 time period. But people didn't understand or could not appreciate what he has done. Then later on, other people try to publish similar type of results. Then they realize, oh, wow, wait a minute. Mendel has already published this. Let's go back and dig it up. And then they found evidence of all these. And he is the one who developed the laws of inheritance. Okay. There are two main laws of inheritance that we will look at. One is the law of segregation. And the other one is the law of independent assortment. And let's go through this one by one. Okay, the law of segregation, it states that the allele pairs, and I will come into what allele pair means, okay, segregate. That means separate during gamete formation. And the paired condition is restored. Paired condition is restored during fertilization. So this is exactly what we looked at in meiosis as to how the gametes are formed, and then they fertilize to restore their diploid status. So what he taught us an allele, like if you have a 2 chromosome, one from mom and another chromosome, a lookalike chromosome, from dad, or a female and male parent, there are specific genes located on these. And so these are the alleles. Okay, there's allele 1, and here's this allele 2 of the same gene. Okay, I would call this 1A, and little, this can be big A or the little a. You would see this type of alleles, and there could be other alleles that are present in these chromosomes. And this is allele 2. It could be, A, or there could be same letter, and we will see how these are going to vary. So this is called the alternate forms of the genes. And so they separate during gamete formation. We saw that the homologous chromosomes separate. And that is what how did he prove this? Okay, so he had plants that are, like, I would keep this as the parental generation. So the parents are a purple color flower and a white color flower. So we have 2 types of, these are called purebred, means you keep on breeding and breeding and breeding for several generations. Everything is purple or everything is white. That's what a purebred means. Okay. So when you get the first generation, which is the F1, like filial generation 1, everything is having a purple flower. Okay. So you think, this
Each
itself shows there's no blending going on. If the blending is going on, you will get a lighter purple, not the regular purple color. And that didn't happen. So it got a purple, all purple flowers. Then he goes to crossing them to the F2 generation. The F2 generation,
each
he got 3 purple flowers and 1 white color flower. Okay, so this is a ratio, rough ratio, and so he tried to explain the way that you're able to get these by giving some notations for the genetic makeup. Okay, so the purple are white color flowers. So these are called phenotypes, the physical trait or physical character or characteristic, they're called. And the white color are purple. So these are individual traits. Okay. So the flower color is the phenotype they are going after, and a specific color become the traits. Okay. So the purple color, he used the notation of, I'm going to use a different color so we can keep track of this. P, that's as a notation, and lowercase p, and this is what is called as the genotype, and the genotype is nothing but the genetic makeup, Okay. So when you cross this, then they got, one, the gamete is going to be uppercase P, and here the gamete is a lowercase p, and they combine together. This becomes one uppercase, one lowercase. You call us a dominant and the recessive. That's the genotype makeup of the F1. So here you need to understand how these genotypes work. So you have the male parent and a female parent. Each of them, when they self, have a dominant and a recessive, and so they get a combination of both dominant or one dominant, one recessive, one dominant, one recessive, both recessive. So all these 3 are going to be the F2 generation. All of these are going to be the purple flower, and this is the recessive, is the white flower. So that is how he explained it, how we got this 3-to-1 ratio. And so if you look at each one of them, how can we explain this? Is you have this both uppercase, you call as the homozygous dominant, and zygous refers to the zygote, the fertilized egg condition. Genetic makeup is same. It's a homozygous dominant, and this one is the heterozygous condition. That's also dominant. So the dominant can express whether it is homozygous or heterozygous, and this is a homozygous recessive. So the recessive trait will express when it is homozygous, and it will not express when it's heterozygous. At that time, only the dominant trait express. Okay. So how did this disprove the misconception that you can see if you have parent, if this is a female parent, this is a male parent, So you have the contribution from both the parents. That's why you're able to restore the white flower in the second generation, and you're able to keep these traits intact. Okay. So after doing this, he defined gene as a unit of heredity, and he did not know what it is made up of, but the meiosis process explained how this segregation of the gametes are going to be formed. So this creates this only one possible dominant gamete, and this creates another gametes, and these are all the gametes in the each generation. Okay. Question so far? Do you all understand the different terminology that I just used? Everybody good? Any question up to this point? Okay, no further question. I'm going to move on to the second law, and then we go on to non-Mendelian inheritance, and we wrap it up. Okay, so the second law states that law of independent assortment. By the way, the first law that we did explain here, this is called as monohybrid. You're talking about one characteristic, and for the second, we are going to go through a dihybrid. Okay, so we will explain that. The law of independent assortment states that the segregation of one allele pair, the segregation of one allele pair is independent of the segregation of other allele pairs. So he treats gene as a discrete unit of inheritance. So he's saying if the flower color is inheriting in one way, the seed color will inherit in a different way. So they are not connected together. So that is called a co-segregation, but here he proves, independent segregation or assortment. Okay. So for that, he uses a dihybrid cross. So this is where the monohybrid cross is a previous one. Dihybrid cross is where he talks about two characteristics. Okay. One characteristic is about the seed color and the seed shape. Okay, is yellow color and round shape is one type. Another one is green color and wrinkled shape. Okay. So the yellow color, the genotype that he assigns to that is this YY, and then this is RR, and this is the lowercase YY and RR. So that is the genotype that he assigns. So when he crossed these two, then as you expect, the parental generation, and that becomes both the dominant and the gametes that this is going to form are going to be, Y and R. So that's a gamete is going to form. Here, it's a Y and R is the other gamete that's going to form. So the first generation, the F1, is going to be yellow and round. Okay, so let's keep this the parents and the F1. And then they self these. When they self these, they get 9 of them. I'm going to give you the ratio of 9 to 3 to 3 to 1. This is going to be yellow and round. So this is going to be a green and wrinkled. And this is yellow and wrinkled. And this is a green and round. Okay, so these are the different types of F2 offsprings that he got. And where did these come from? Okay, so you need to understand 2 things. One, the seed color, yellow or green, did not go always with the seed shape, round or wrinkled. So these 2 are parental. Okay. So this is also parental. They look like their parents, whereas these 2 are called as recombinants. So they have a combination of 1 dominant, 1 recessive, and so on. So if you follow this, this is going to have a combination of YY and RR. That is a genetic makeup of the F1. So in this case, you need to figure out, how all these things are happening. Okay. So let's use process called as the rule of multiplication. Rule of multiplication. Okay. So the case of yellow and round, the chance of getting an yellow is 3 out of 4 is yellow, and the chance of getting round is also 3 out of 4. That's both are dominant. So from the previous example, we saw the chances of getting a dominant at 3 out of 4 is equal to 9 out of 16. That's how you got this yellow and round. Okay. And the chance of getting a recombinant is, 3 out of 4 is either yellow or being round, both this characteristic. And 1 out of 4 is going to be the recessive, which is either a green color or wrinkled shape. Okay, so this is a combination they are getting. So that is 3 out of 16 in each cases. So that is what creates this 3 out of 16 in both cases. That's 3 out of 16. Then the chance of getting a recessive is 1 out of 4 times 1 out of 4, which is the green color is the recessive and wrinkled shape is the recessive, and that is 1 out of 16. That is what this ratio is. Okay. Everybody good so far? So you can use this rule of multiplication by understanding what are the chances of getting yellow and round that separately from yellow and wrinkled or green and round and so on. So this proved that the segregation of seed color is independent of the segregation of the seed shape. Okay. So this is not Hardy-Weinberg equation, but this is a basis of Hardy-Weinberg equation. What do you say is correct? This is related to Hardy-Weinberg. Okay, good point. Yeah, this is the basis by which they have X percentage of this allele, Y percentage of that allele. So that's a percentage, and this is where the starting point of Hardy-Weinberg. Okay, good. Any other question? Hold on. Yeah, I can hear you now. Go ahead. Somebody speaking? Any question? I see somebody unmute. Nope. Okay, that's fine. I'm going to move on to the last part of this is non-Mendelian inheritance. So these are cases that Mendel did not figure out any of these, but it happened later on, and non-Mendelian inheritance is other types of patterns that other scientists discovered after Mendel published all these, and that's what it's called, non-Mendelian inheritance. Okay, so you don't need to do this patterns there, but understand how this non-Mendelian inheritance work. And number one is incomplete dominance. So this is not, one characteristic is not dominant over the other characteristics. So here the flower color in snapdragons and carnations, many of these that you would find. So when you cross the red and white color flower, so it will generate a pink color flower. And you go to the next generations, you get one red, two pink, and one white. So this is considered as incomplete dominance. Red is not completely dominant over white. So this is a parental generation if you assign the genotype, and the heterozygous condition provides the pink color, and the homozygous dominant is the red color, and the homozygous recessive is the white color, and this is the heterozygous conditions are going to be the pink. So the intermediate inheritance doesn't mean that this is a blending going on here. Okay, there's no blending going on because you're able to restore the white color in the F2 generation, means this not blending, you're not getting a lighter red or pink color in the middle. So that is the point of this incomplete dominance. And the second one is called codominance. Codominance is where one allele is not dominant over the other allele. Say, for example, the blood type. We have the blood type A, the blood type B, and the blood type, AB, and the blood type O. So these are determined by alleles called IA, IB, or I, which is no A or B in there. That's what creates the O allele. And based on the combination of A and IA, and IA will create this. So let me give you both possibilities. So this can be an IA, or IA and I. Both are create the A. And the second one can be an IA, IB, and IB, or it can be IB and an I, that's an allele. And the AB is and simple and IA, IB, and the O allele is nothing but I and I. There are no proteins formed by that. So that is A is not dominant over B, B is not dominant over A. That's called a codominance. Okay. The third type is called as epistasis. So epistasis is one allele pair, okay, affects the expression of another allele pair. Okay. So in this case, the good example is the fur color of the golden retrievers. You have a black or brown color, and so the you can get an yellow color retriever, which is an absence of the gene to express the pigment black or brown on the fur. So this is due to the lack of the expression gene. That's what is called as an epistasis. And we don't have to do the Punnett square or the allele distribution for this. And then go into the next one is called pleiotropy, where one gene defect has multiple phenotypic effects. So before we saw this flower color, seed color, seed shape, they're all one allele affect one characteristics here. One defect, multiple effect. The examples is sickle cell anemia. Just one gene mutation makes a huge difference. Also, cystic fibrosis. There's a one gene defect in a chloride ion channel in the lung makes a big difference in there. Okay, and in addition to this, you also have epigenetic, which is outside, beyond one's genome. So this is again how the external factors affect the gene expression. Affect the phenotype. So this is all we are trying to understand on all these things. And then you also have the environmental effect. Environmental, which can be the nutrition and the exposure to the elements, which can be light, heat and pollutants and things like that. So this is all external exposure. All that is considered as non-Mendelian type of inheritance. So these are some examples all that you need to understand what each of these mean and what are the examples for them. Okay, that's it. Any question up to this point?